Okay, hello everyone. It is the evening of October 26th, a Monday evening. Hope this recording finds you well. Um, wow, we are well over halfway through the semester, and this is about the time, if we were in person, I would tell you to pat yourselves on the back because for many people, for those who've trotted this path before you, they generally tend to agree that the second unit of the class is the most demanding. So, you've made it through the worst, according to popular opinion. So pat yourselves on the back. Just some announcements before we get into lecture for today. Exam 2 is graded, so you should see um, the um, the exam graded on your grade portal on Canvas. There are a couple of folks who had good reasons, and I gave extensions to. I haven't gotten rid of those yet, but um, generally, great job. I was really impressed. So I don't have any means for anyone, but great job. And so take a look at that and let me know if you have any questions. Please email me if you have any questions. Just a reminder, this is an online class, and maybe you can tell I do not like to be the bad guy. In instances where maybe you forgot or something like that, the syllabus has a policy where we can't take makeup exams unless there's an emergency, like a health issue. That's pretty much the only situation. Um, for exams, generally speaking, I put them on Canvas relatively shortly before they're due, and Canvas won't necessarily send you a reminder. So the syllabus is absolutely your best guide for the class, and the syllabus is that document right there on the top of the Canvas page. It tells you where and when everything is happening. And also the announcement slides here in the lecture are where I'll generally tell you any kind of changes that are going to go on, and I'll also remind you. So please don't rely on the Canvas reminder system. It's not bulletproof. Rely on the syllabus. That's the ultimate guide for the class. The syllabus is the one that's in charge. And also, look at class emails and the announcement slides here that I try to do before getting into a lecture for every recording. So just something to keep in mind. Exam 2 is graded. Analyzing my phenotype, the lab, is now on Canvas, and so it's a little bit different this week because it's a worksheet. It's not something that it's not like an instruction document as it has been in the past. You can feel free to print that out and do your lab directly in that worksheet. I implemented it off of what a couple of my colleagues did. I just think they did a better job than doing it if I were to make my own lab for you. You can absolutely still write it in your lab notebook, or you can print it out and scan and write in and scan it back. It's up to you. One thing about the lab is that it does ask you at one point to find a partner to simulate a reproduction event where you find a partner and you have a paper bag, and you exchange gametes and your offspring. Then it's basically an exercise in heredity. So read the document, but I just want to emphasize, because we're in the middle of a pandemic, this is not an expectation that you go out and to somebody's house that you haven't been around in a while. The general expectation is that you do this exercise. It should be a fun exercise with someone that's in your household, whether that be your significant other or family member, read the instructions. It's pretty simple, but it does involve you like sitting at the same table or something and talking. So I don't want your health to be at risk as a result of this. So keep that in mind for this week for the Analyzing My Phenotype lab. The rest of it is independent. You could do it on your own. So today we are actually starting Unit 3 of the course, which you'll notice is a much shorter unit. We only have 3 lectures. It's far more conceptually based as opposed to memorization, which, speaking for myself, I'm a conceptual person. I do not like having to memorize and keep a bunch of bits of information in my head. And so it's a shorter unit on purpose. We only have 3 lectures and 2 labs in this unit. And so just to give you a timeline, we're doing inheritance and heredity today. And then on Wednesday, we're going to do DNA structure and function. Next Monday, a week from today, we're going to do DNA technology. And then next Wednesday, we're going to have another review session. And I'll probably put out another poll for when the review session would be, when most folks can be able to attend. But just keep that in mind. I mean, basically this week and next week are the content aspects of this unit. And then exam 3 is coming up in a couple of weeks. So I know that's a pretty quick turnover, but again, this unit is conceptual. And you've probably heard of some of these concepts before as well. So just so that's on your radar for this Unit 3. Okay, so just as a reminder for the trajectory we've been taking into this class, in Unit 1, if I can hearken your attention back, we talked about ecology. So populations and communities and ecosystems and evolution, and the various processes that impact how ecology is studied as a science. And we talked about some of the core principles of ecology. Then in Unit 2, the unit we just got done talking about, we completely started at the base, literally. And we broke life down into its base chemical constituents. And we talked about biochemistry, and we talked about how these bonds can arrange themselves in together to form molecules that eventually form bigger molecules, these macromolecules, and how those macromolecules can themselves form together to form organelles and ultimately cells. And then we talked about how cells can function and what cells are what their internal arrangement is made of. So that was Unit 2 of the course, which we broadly called microbiology, the study of small things. Okay, so Unit 1, then Unit 2. This current unit of the class is genetics. Okay, so that's Unit 3, and it doesn't really exist, at least visually, anywhere here on this continuum, because really genetics intercedes every aspect of this schematic of life. Okay, so it really is interwoven throughout all of this. I can't pinpoint a specific spot on this schematic where genetics exists, because it really intertwines everything. So we're studying genetics in this unit, which is the study of heredity and gene variation in organisms. Okay. And specifically today, we're going to study inheritance and heredity, which is found in chapter 9 of your textbook. So just some terms that are really important to get down before we really delve into the subject matter of this class, and some of this lecture. Some of these you've heard of before, others you haven't, at least in this class. Others, even though we may not have talked about them in the class so far, you've probably heard them used in different contexts, maybe when you took general biology in high school, or if you've taken another biology class somewhere, or just colloquially out in society. You've probably heard of some of these terms before. But let's just go ahead and define these terms officially, because in order to proceed through the rest of the unit, we have to be able to use these terms more or less easily. And so let's define them so that we can use these terms in a fairly, after this slide, I'm going to assume that you know what these things are. Okay, so one by one, if we say the word chromosome, the word chromosome, when we say chromosome in the singular, not the plural, but a single chromosome, is simply one DNA molecule. Okay, it's a single structure of DNA with all of its composite proteins that it's kind of wrapped around. Okay, so here's a nucleus inside of a eukaryotic cell. Okay, and inside of an individual nucleus, there can be one to many individual chromosomes, but let's just take one chromosome in particular. Okay, one chromosome, right, like this is an artist's rendition of one, is simply a packaged form of a single DNA molecule. So it's a very, very highly organized, highly wound up single DNA molecule that are wound around these proteins. So a chromosome is DNA and protein, but it's a single molecule of DNA. Okay. Getting a little bit more specific, when we say the word gene, a gene is simply a section of DNA along a chromosome that codes for something. Okay, and we'll talk about how this process works a little bit later on in the unit in a couple of days. But a gene, if we literally say what a gene is, it's simply if you pick out a specific stretch along a DNA molecule, as shown here by these brackets, a gene is simply a stretch of DNA molecule that codes for something specific, like a specific protein, for example, which is the basis of gene expression. Okay, so on an individual DNA molecule, you might have many, 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 many genes. Okay, but a gene is simply a stretch along the DNA molecule. Okay, getting even more specific, an allele is simply a specific version of a gene. Okay, so a gene is a stretch along a DNA molecule, and an allele is a different version of a gene. So you might have a gene that codes for proteins that affect your eye color. Okay, so that's the gene that affects your eye color. But there are many eye colors, of course. So you might have an allele that codes for having brown eyes or blue eyes or green eyes or hazel eyes. Those are different alleles, but they're all the same gene. In other words, eye color. Okay, a change to a gene is a mutation. And relative to like hands sprouting outside of your chest or you growing a third armpit or something, that's kind of what a lot of people think of as a mutation. That's not what a mutation is, as you can imagine. We'll actually officially show what a mutation is when I say a change. I'll describe exactly what a change to a gene is, but a mutation is simply a change to a gene. It doesn't necessarily mean anything bad. It can be bad, but it can also be good. It just depends. And then a particular set of alleles for an organism, if you combine all the specific types of alleles, the specific version of alleles for all your genes that make you as an individual, that's your genotype. Okay, everything that makes you genetically unique, that is your genotype. And if we think about the actual physical expression of your genotype, like the protein expression of your genotype, your physical appearance, that is your phenotype. So genotype is literally the vast sum of your alleles, and your phenotype is the physical expression of your genotype. Okay, so I'm sure you've heard these different key terms before in different contexts, but we have to be able to use these shorthand from here on out in order to discuss the rest of the concepts in this course. Okay.
I had a question.
Mm-hmm.
Um, um, could different genotypes have like the same outcome? Like say like would different could different genes have like result in like the same color, like if like hair color or eye color or
That's a good question. We will talk about that a lot actually in different types of gene expression. genetics is crazy, wacky, complicated. And so when you talk about different genes interacting with one another, like the genes that code for your hairline, for example, the genes that code or the gene that codes for your hairline can interact with the gene that codes for the color of your hair or your eyebrows, for example. And so your genotype is think of your genotype as like, split pea soup. It's the mixture of all the different things that make you genetically unique. And that can mean a lot of different interactions between different alleles, different versions of genes. And so what that means in terms of your phenotype is highly context dependent. And we'll talk about different examples of how that can occur. But yeah, a good question. We'll go through some specific examples. But this is still, I mean, this unit, more than anything else in the course we talk about, genetics is still a hot topic in the field of biology. And so some of these things are still not resolved. We're gonna kind of come close to the cutting edge of what biology in terms of genetics has been able to discern. And so your question is a good one, but there's aspects of it that are as of yet still unknown, which is a pretty interesting thing in and of itself. So the answer is to your question, it depends. It's complicated, but still an area of active research.
Well, it's good to know that they're researching it, at least.
Oh yeah, yeah. There's at least one in every major university. There's people who study genetics all the time. It's pretty commonplace now, actually, for people to study genetics. I had to take a genetics class in undergrad. I'm not a geneticist, but it's still an important discipline, and it's very relevant in our society today. There's lots of different implications, even for society at large, and we'll go through some of those examples. Okay, so I don't think I need to convince you, and you probably know this already, that DNA is the basic unit of heredity. Okay, this is kind of one of those colloquial shorthands. You know what DNA is, I'm sure, before you got to this class. And we'll talk about what DNA is specifically in the next unit, but I don't think I need to convince you that DNA is the basic unit of heredity, the molecular unit of how uh, uh, genes and alleles and genotypes and ultimately your phenotype, the end result of gene expression, all of that has its basis in this really amazing molecule called DNA. And we've talked about how DNA lives in the nucleus of eukaryotic cells. So here's a cross section of a nucleus in a eukaryote. And we've talked about how it can take on different forms depending on the phase of the cell cycle that a cell's in, from existing dilute inside the nucleus during the interphase section of a cell life cycle, this chromatin structure, which is basically dilute DNA. It's kind of like spaghetti. But then when a DNA is ready to divide and transition into M phase, which we talked about mitosis, it has to package itself up because it's moving house. And so that's when you get these really intricate structures of DNA wrapped around these proteins called histones. That's amazingly efficient, wrapping of DNA around these proteins. And ultimately, when it's wrapped all together into a nice neat bundle, that is called a chromosome. An individual DNA molecule is hecka long. We're talking about your DNA, every single one of your cells has millions of DNA molecules, or your body rather has millions, billions of DNA molecules. Yet each individual DNA molecule is long enough to go to the moon several times over. It's just really long. And so if you think about something that's long enough, if you stretch it out end to end to go to the moon back and forth many times, and yet you can still package it up so efficiently that it's in a structure called a chromosome that's smaller than the nucleus of a eukaryotic cell, many times more fine resolution that you can make out with your naked eye. That's just amazing. And so the chromosome is a wonder of engineering in and of itself because of how efficiently it wraps up a DNA molecule. But I digress. DNA is the basic unit of heredity. And another thing that's really interesting to point out from a historical perspective It's that DNA, even though we talk about it so easily these days, DNA is everywhere. You've probably heard it before. It was discovered pretty recently within living memory. Maybe you've heard of these two gents, James Watson and Francis Crick. Crick was a British mathematician, and James Watson, who's still alive, although he's pretty old now, he's in his 90s. He's an American. He's actually an entomologist, so he studied insects. These two guys teamed up when they were at Oxford in England, and there's a bit of a complicated history there, which I won't get into. It's a little bit murky, but to spare you the details, in 1953 they came out with a really seminal paper that described the DNA double helix as the actual chemical structure of DNA. Here's actually their model here. Crick, again, was a mathematician. He was a modeler, a very bright guy. And he, through really elegant mathematical formulas, was able to calculate bank angles and all these different things and really describe the structure of the DNA molecule, this double helix. Okay, so this was only in 1953. Okay, really not all that long ago. Okay, when my father was going through undergraduate degree, he was born in 1947, and he did not learn about DNA when he was a biology major in undergraduate at Ohio State. That was in the 60s. It even took some time for the paper to get some traction. And this paper, it kind of very humbly posits this molecular structure of nucleic acids, which again we've talked about before. It's only 2 pages long. It's amazing. Length does not necessarily equate to quality. So this paper is 2 pages long, and yet it's so seminal. It's on the order of The Origin of Species by Darwin. That's how important this paper was. It's influenced a lot of biological research for the latter half of the 20th century and well into the 21st century. The genomics is the field that we call it today. Bioinformatics also. Very, very phylogenetics is another term. Very, very wide, very complicated fields in and of themselves with multiple subdisciplines now. And yet it all harkens back to this paper in 1853 by Watson and Crick. So it wasn't all that long ago that we did not know what the chemical structure of heredity is based on. Okay, but even if we go back a little bit further than that, even before we understood what the chemical structure of DNA is, we've, as human beings, have known for millennia that we have some sort of heredity. If 2 people have a child together, it's pretty clear that the child often, generally has traits, a phenotype that is a combination of the mother and the father, be that for humans or for animals or even for plants. Okay, so we've known about heredity a lot longer than we've known about the chemical structure of DNA. And one of the, going back to, ancient times, even prehistory, this is a known phenomenon. But one of the first people to actually apply the scientific method to the study of heredity, and maybe you've heard this name before, was a guy by the name of Gregor Mendel, who was an Austrian monk. Okay, so he was a very devout Christian in the 1800s. This is his actual monastery here in I think it's in Prague. I'm blanking now, actually, which is embarrassing because I actually went there as an undergraduate during my semester abroad. This is his actual pea garden. You can actually still go see it today. They kind of keep it the way it looked when he was a monk at this monastery. Okay, and so he was the first person in his spare time. This was how the monastery fed itself, by growing peas, amongst other things. But he was the first person to apply the scientific method to the study of heredity inheritance. And so, his work on pea plants in this garden monastery garden was where we really deduced the fundamental principles of heredity. Starting to lay down some laws of heredity, and this again predates the actual chemical understanding of what DNA is. So this was before we even had the term DNA. We knew at some point that there was some biological mechanism, even though we didn't know what it was. We knew that there was some kind of biological mechanism regulating heredity. Okay, so Gregor Mendel was the first person to do that in a robust way, and he, of course, studied pea plants. Why did he study pea plants? Well, there's a variety of different ways and reasons, but here are just a few really good reasons why plants are really model organisms for studying heredity. For one, it's super easy. I'd wager you have a garden at home or you've grown plants before. I mean, we live in the San Joaquin Valley, for crying out loud. Agriculture surrounds us in every direction. Okay, so plants are relatively easy to manipulate, and we do it all the time for our existence. We wouldn't be able to survive without it. So, all you have to do in order to study plants and their heredity is get some plant seeds and plant them in the ground and grow them until they produce flowers, assuming these are flowering plants. And then instead of allowing the plants to do their thing and reproduce sexually on their own, you hand pollinate the flowers by doing things like removing the male reproductive parts with a pair of scissors, so that they're not spreading their genes to surrounding individuals, and instead taking something like a paintbrush from one plant and artificially inseminating the female parts of another plant. Okay, so this can be done pretty easily. So by hand pollinating flowers, you can actually control, you're basically taking the role of a pollinator like a bee, and you're actually intentionally able to control which organisms, which individuals are exchanging genetic information with others. And so, so plant A of this phenotype, which in this case is purple petal color, you could cut its stamens and then paint the pollen on the female reproductive parts of this flower, which is white petals, and you can use that to basically control heredity. You're taking the role of a pollinator. So you do this, and then you collect the seeds. Okay, so this plant is now pollinated. It's a pea plant, of course. So it's going to produce a nice pea pod with peas, with little baby beautiful peas inside. Aren't they cute? And you collect those seeds. Okay, and then you do the same thing the next generation. You plant those seeds and you look at what their phenotype is. These are the offspring of the ones that you controlled artificially in the first generation. Okay, this is called the F1 generation, the first cross, the first filial cross, as it's called. And then you can do that again and again and again through multiple generations. The next generation after that, the grandchildren are F2. The generation after that are F3. Okay, and you can do that again and again and again. Plants have relatively short lifespans, annual plants that is, and you can do this many times, even within an individual growing season. So plants are relatively model organisms. Annual plants are relatively model organisms to study heredity in. This is exactly what Mendel did. He did exactly this, as shown in this schematic right here. Okay, to backtrack a little bit, we talked about mitosis, which is a phase of the cell life cycle, which is a form of asexual reproduction in cells. Okay, so a lot of organisms reproduce asexually, so they do not exchange genetic information. They basically clone themselves. So plants are known to do this. Joshua trees, for example, are known cloners. The quaking aspens are known cloners. Even some animals are known to do this, like copperhead snakes. Female copperhead snakes can basically clone themselves and give birth to live young that did not require insemination of sperm by a male copperhead snake. Worms. I don't recommend doing this because it's kind of cruel, but worms, annelids, You can take a worm, generally speaking, and if it's a healthy and robust worm, you can pull it in half, much like sea stars. And those two halves can often heal off and become functional individuals. All of this is the product of merely mitosis, not meiosis. And so you get clones that are identical to the mother. Okay, no exchange of genetic information. There are a variety of reasons for why organisms might choose to reproduce this way. It's quicker. It doesn't involve the production of costly gametes, which are energetically costly to produce. You don't have to go through all the energy of finding and maintaining a mate, which has its own slew of energetic costs and behavioral costs to it. So for a variety of reasons, some organisms only reproduce asexually. But there are shortcomings to it, which we'll talk about a little bit later on in detail. Most organisms on planet Earth still go through the effort of reproducing sexually, which means literally the exchange of genetic information. We do it, pretty much all, with the exception of relatively few cases. Most animals do it. Most plants do it. Microbes even do it. So, sex is common. And why do it? Despite the fact that it's really energetically costly. Flowers, for example, are a huge energy sink for plants to produce. They're beautiful, of course, flowers, but it costs the plant a lot in order to produce flowers. And the only purpose for flowers is to attract pollinators and get their gametes out there right front and center of potential pollinators so that genetic sexual interaction can occur relatively easily. But it takes a lot for plants to produce that. Why go through all that effort? Well, it creates genetically diverse offspring. Okay, so the offspring of a sexual reproduction event have more genetic diversity than the offspring of a asexual reproduction event, and there can be a whole slew of advantages to that. More resistance to disease, for example, more resistance to stress, for example, a greater ability to adapt to changing environmental conditions, for example. So there's a whole slew of potential advantages that an organism that's the result of sexual reproduction can have over an organism that's the result of clonal reproduction. Okay, and we've also talked about biodiversity, harkening back to the first unit of the class. We talked about biodiversity, which is basically, it's a really broad term that's a metric of the variety of life in a particular habitat or ecosystem. And there's a bunch of different ways biodiversity can be studied. It's really a loaded term because there's so much to unpack in the context of biodiversity. It doesn't mean just one thing. But one aspect, and one really important aspect of biodiversity is genetic diversity. And remember, go back and watch that lecture where we first unpacked the term genetic diversity. We talked, this is the same slide from that same lecture. I said genetic diversity is a component of biodiversity, but we're going to explore it later. Well, now is later. Now we're actually going to just talk about genetic diversity and as a component of biodiversity. And next slide. Okay, so where does genetic diversity or genetic variation originate in the context of sexual reproduction of organisms? Okay, there's two broad ways that I'm going to posit to you that genetic diversity or genetic variation actually originates at the genetic level. The first we've already been talking about, fairly obvious, sexual reproduction as a result of meiosis, which we've talked about in the last unit, and recombination. That's a huge way, really the majority way that genetic diversity originates. It makes sense, of course. Genetic diversity is a result of sexual interaction, sexual exchange of gametes. But it's not the only way. Interestingly, as it turns out, mutations, which remember we defined mutation as simply a change to a gene, mutations are another pretty significant source of genetic diversity, genetic variation, and we'll talk about what a mutation is. Since we've been sitting here, we've had mutations going on in our bodies, and that can contribute to genetic variation as well. That's quite distinct from sexual reproduction. Okay, so this is the same slide you've seen before in the context of genetic variation and genetic diversity. That we're talking about meiosis, and we didn't unpack meiosis in any great detail. Suffice it to say that it's the type of cell division that cells go through when they are producing gametes or sex cells, which, as you can imagine, is pretty crucial for sexual reproduction. Okay, and one aspect of meiosis is this really interesting phase, which we'll talk about in the next slide, actually. But the basic goal of meiosis, and this again is distinct from mitosis. Meiosis is sexual in nature. The goal of meiosis is to produce gametes, okay, which are sex cells. So let's talk humans, for example, because we're humans and it's easiest to understand in terms of something we understand. When you have like the cells in your hair or your skin or wherever, your typical cells in your body, those are what are called somatic cells, meaning that they're cells that are not involved in meiosis. They simply, for the most part, go through mitosis. Those cells, those somatic cells, have a full complement of DNA in them, meaning they have all the DNA that they need. Okay, those cells, those full complement of DNA cells, so somatic cells, those are called diploid. Another term is 2N, diploid and 2N, basically meaning two nucleosomes. Di meaning two, and ploid meaning ploidy, which is another term we'll talk about here shortly. But the vast majority of all cells in your body are diploid. They have a full complement of DNA. But if we talk about cells that are going through the process of meiosis, so the process of producing gametes, that's a slightly different story. Okay, so a cell that has gone through the process of meiosis, which is basically mitosis times 2, so it produces 4 daughter cells as opposed to 2. Okay, it's gone through mitosis twice in a really broad way. And so it produces 2 cells, much like this, okay? But then it goes through that process again, and it produces 4 cells. And during that second phase of meiosis, as it were, the actual DNA, the complement of DNA inside these cells is split in half, okay? And so the result is that the genome of gametes or sex cells have a half complement of DNA. And this is pretty easy to know. That's the term haploid. So I think of it in terms of half, meaning half. And they have a half complement of DNA, or N, simply N. So diploid or 2N those are your somatic cells that have a full complement of DNA, and haploid are your gametes, and they have a half complement of DNA, or just simply N. Okay, so whether it's in the case of humans, egg cells for females or sperm cells for males, human beings have 46 chromosomes in a typical somatic human cell, but for haploid gametes, whether that be eggs or sperm, the number of chromosomes in these gametes is 23, half of 46. That's why they're haploid. Okay, they have a half complement of DNA. And then there's the full process of fertilization, which of course this is not a sex ed class, so we will not get into the gory details. But that is the cellular basis of sexual reproduction in the context of meiosis. And we've talked about this really important step in meiosis. This is probably the most important step in meiosis. It's the real point of it. So somewhere between, in the context of prophase 1, there's this really important step that occurs. It's a fairly obscure step, but yet still important, called recombination. And we actually talked about recombination very briefly in Unit 2, but we didn't go into very much detail. But here it is right here, shown in this schematic right here. Recombination is this step in meiosis where literally the chromosomes from 2 individual parents, from mother and father, they go through this what's called the recombination event. Some people call it crossing over between non-sister chromatids. It means the same thing. So here it's shown right here, two separate what are called bivalent chromosomes, but they go through this process of crossing over, and as a result, the two chromosomes literally exchange sequences of genetic information. The result is that when meiosis, the process of meiosis is complete, you get these four separate daughter chromosomes that have completely novel sequences of DNA. And so this process of genetic recombination during the crossing over event is sort of the crux basis of where genetic variation in the context of sexual reproduction occurs. These are entirely new and they're inherited by the offspring. Here's an electron micrograph of two bivalent chromosomes crossing over in a recombination event. So this is very important, okay? So you and I are the process of many, many, many recombinations, and that's what makes us us as individuals. It's really where our genotype emerges, and that's what makes us unique. Okay, so sexual reproduction as a result of meiosis and recombination, that of course is a very important way that, where the vast majority of genetic diversity originates. But there's another way, as we've already discussed, that is not insignificant. It's a huge way that cells, as it turns out, and genes diversify themselves, and that is mutations. And this is not sexual at all. Okay, so we talked about mutations being a change to a gene. Okay, so that's the base definition of what a mutation is. It's not, arms spreading out of your head or zombies taking over or anything like that. Some mutations can be bad, but an individual mutation in singular, one single mutation event is simply a change to this right here. Okay, so I'll harken back to the structure of nucleic acids. Nucleic acids, of course, are one of the four types of biological macromolecules. And we talked about, not in a great amount of detail in Unit 2, but we talked about how nucleic acids have a number of different subunits to them. They have a ribose or a deoxyribose, depending on what type of nucleic acid you're talking about, shown right here, this nucleotide. They have a phosphate group, and then each individual nucleic acid has a different nitrogenous base. And we'll talk about these different types of nitrogenous base a little bit later on in the next unit, or in the next lecture in the class. But for now, all I want you to keep in mind is this base right here. And just think of it in terms of an A, a T, or a G, or a C. Okay, so A's, T's, G's, and C's are very important. Again, we'll talk about the actual structure of DNA in the next unit, so we're only glossing it over it here. But we're talking about a change to an individual nitrogenous base. That is a mutation. Okay, so it's happened probably 1,000 times in my body since I've just started this slide. It happens all the time. An individual mutation is simply a change to a nitrogenous base in a single nucleic acid. It happens a lot. Okay, case in point, mutations mostly occur in the context of DNA replication for when your cells are actually going through mitosis or meiosis. And so here is an actual DNA molecule. We're not going to go through the process of how this works, but because it's fairly complex, it involves a number of different proteins that kind of unzip a DNA molecule, and these proteins have a variety of different functions. But these bases here, see these lines here that connect these two helixes of a DNA molecule? An individual mutation is simply a change to one of these little things right here. Okay, so every one of your DNA molecule has millions to tens of thousands to millions of these individual nitrogenous bases. It's the actual code of DNA. And so when one single nitrogenous base changes, that itself is a mutation event. That's a single mutation event. Okay, so there's a number of different types of mutations. And again, all I want you to keep in mind at this point is A's, T's, G's, and C's. We'll talk about what these are in the next lecture. Okay, but we're actually looking at the, in half of a DNA molecule right here in these two schematics. Okay, so keep in mind, a mutation is simply a change to a gene. It's a change to a nitrogenous base. So that could take on a number of different forms in the context of DNA replication. Okay, one is that okay, imagine this protein coming along here, and this protein's job is to insert the next DNA nitrogenous base along the line. Okay, but let's say that that protein makes a mistake. Okay, let's say it accidentally puts one, let's say it puts a C where there should be a T. Okay, so it substitutes one nitrogenous base for another. So here's the original sequence, but this protein's coming along. But oops, instead of putting a T, it puts a C. Okay, so that's a point substitution. That is one type of mutation, and that mutation has just happened. Okay, arms are not sprouting out of your chest at this point. Okay, this is not what's happening. Another type of mutation is duplication. Okay, so this is another type where a protein can conceivably make a mistake. So it's coming along, and instead of putting in an extra base or putting in switching in one base for another, it accidentally deletes or duplicates rather an individual nitrogenous base. So it's going along here and it puts a second C base here, and so it shifts the whole DNA molecule that way, which is called a frame shift event. It's a duplication of an individual base. Okay, that's one type. And the third type we'll talk about is the opposite. This protein's coming along and it accidentally leaves a protein or a nitrogenous base out. Okay, so instead of putting this T base here, it leaves it out and it frame shifts that way. Okay, and so point substitutions, duplications, and deletions are three types of mutations. And a single mutation event is simply a change to one single nitrogenous base. It happens a lot, okay? But if it happens a heck of a lot, I mean, millions and millions, okay, I'm being a little bit facetious here, but if it happens thousands to millions of times, it just depends on the organism, depends on the gene, whatever the case may be. If this happens a lot, that's when you may get a change to a phenotype. Okay, so remember, the genotype is just the vast sum of your genes, your different alleles, and mutations are changes to the nitrogenous base in a single gene, and your genotype is the vast sum of your genes. Okay, but and mutations happen fairly common. It happens in DNA replication. It's not unheard of. It does not mean that arms are spreading out of your chest. But if it happens a lot, a heck of a lot, that's a contribution to genetic diversity. That's why mutations are a source of genetic diversity. And if it happens to be on a certain degree, that may change the phenotype, maybe. Okay, and that can result in new sequences of DNA and maybe even new species coming onto the scene without any kind of sexual reproduction. Okay, so as I've already been hinting at really strongly, most mutations are completely benign. It happens in your body every day, an individual mutation. Okay, it happens all the time, and it does not change you at all one iota. Okay, it doesn't impact your protein function at all. Sometimes mutations, if they add up beyond a critical mass, they can be bad for you, and there are genetic diseases that are a result of mutations. Various genetic we'll talk about actually some examples of that, of mutations that can be bad. Yeah, they can kill you, but again, this is pretty rare. Interestingly, this kind of gets back to the how sometimes terms can be misleading. We often think of mutations as being a bad thing. Sometimes mutations can actually confer evolutionary fitness in organisms if they happen to, let's say a mutation, mutations occur beyond a certain critical mass and a new phenotype emerges for an individual that's different from all the other organisms, the individuals in that population. Um, let's say that mutation actually confers some sort of evolutionary advantage. So let's say through the process of mutation, a person is born who's just extra smart. I'm just being a little bit facetious here again, but someone is born a genius as a result of some kind of mutation. They're a prodigy. That's an evolutionary advantage, and maybe that becomes selected for over time, and it becomes a more common uh, thing in the gene pool as a result of it being advantageous, because that individual organism with that mutation is more successful at producing offspring. Okay, so I digress a little bit. I'm getting a little bit ahead of myself here. Mutations can happen in all cells. Okay, somatic cells as well as gametes, but they're only passed down in gametes. Okay, so for example, here on the left we have an apple that is has a mutation of two different cultivars. So like Honeycrisp is 2/3 of this apple, or about half of this apple, whatever you want to call it. But it has a mutation where whatever happens, the genes that are influencing this particular cultivar of apple, it looks like Red Delicious on one side and Honeycrisp on the other, if you know your apple varieties. And so this is purely a somatic mutation. It's, you might, maybe you'll see this at the grocery store sometimes. Generally, grocery stores weed these type of produce out because they don't want to freak people out. And it actually cuts into their profit margins, apparently. But, you can eat this apple and it absolutely will not hurt you. I bet it's delicious. But if you take the seeds out of this apple and plant them in the ground, it's very unlikely you'll get an apple that looks like this, because this mutation is somatic. It does not exist in the gametes. And in order for a mutation to be passed on to the next generation, the mutation has to exist in the gametes. So, for example, one sexual mutation is this tendency of fascication, which occurs in a lot of plants, but it's pretty common in the sunflower family. So here's a a healthy sunflower. I'm tempted to tell you what a sunflower is right now, but I have to stop myself because that's a huge divergence. This is not one flower. Okay, I'm going to go ahead and tell you. Each petal is a flower, and each thing in the middle is also a flower. So this whole thing is actually many, many, many flowers. But I digress. Sunflowers tend to go through this process by which they can fascicate themselves. It's a sexual mutation where they can, basically stack different discs and rays many, many, many on top of each other. And that's, again, a sexual mutation. If you take the sunflower seeds from the sunflower and plant them in the ground, it wouldn't be entirely surprising if some proportion of the offspring have have this fascication. Okay, and I think I'm going to go just a little bit further before I'm going to cover these next few slides, because I don't want to make this lecture too long. I'm going to carry on the rest of this on Wednesday. But okay, so we've covered mutations in some detail now. Okay, so let's go all the way back to Mendel. Okay, Mendel was that Austrian monk who was studying pea plants in his monastery garden in the 1800s. Okay, so he was artificially working with these pea plants and hand pollinating them across multiple generations and keeping track of what the phenotype of the generations were. He did this for many, many, many years. He actually did not get recognized with this in his lifetime, but he got recognized after he died. But he did this for many, many years. He kept very detailed notes. He had a biology notebook, which is another example of why biology notebooking is such a valuable exercise to go through. Anyway, so he had these pea plants in his garden. He was collecting really robust data. The end result of his really laborious efforts, he came up with these hypotheses on the basis of the patterns that he was observing. And we call these the Mendelian hypotheses or Mendelian genetics. So these are the base laws of genetics that Mendel first posited. Okay, so let's break it down. And maybe you've heard of some of these before. You probably would have heard of these when you took general biology in high school. So first of all, when an organism inherits two versions of a gene, so one from each parent, okay, so let's think of, let's say your father had blue eyes, but your mother has brown eyes. Okay, so those are the same gene, but different alleles. Okay, so one gene from each parent. When the organism, the offspring, in other words, is homozygous, if both of the genes for that allele are identical. Let's say both of your parents have brown eyes. Okay, you as the result of the offspring of your parents are homozygous for that gene if both of the alleles that you carry for eye color are identical. However, if you carry different versions of the same gene, you are heterozygous. Homo meaning the same and hetero meaning different. So the organism is heterozygous if the different alleles are different. So let's say your father has blue eyes, your mother has brown eyes. You're born. You have certainly one eye color versus another, but you're heterozygous because both of your parents had different versions of the allele and you carry those versions. Okay, so getting a little bit more specific, if you are heterozygous for a given a gene, so you have two different versions of an allele of the same gene, always, at least in the context of Mendelian genetics, one allele usually dominates the other. We call those dominant alleles, and those are the alleles that determine your appearance. For example, my father, I look like my father. He brown eyes, brown hair. My mother has red hair and blue eyes. So both of those are recessive traits. I got the dominant traits, and my sisters as well. We all have brown hair and brown eyes. Okay, so the gene that codes for brown eyes, that allele is dominant over blue eyes. Same thing with kind of brown hair over red hair. Those are recessive. Okay, where generally speaking, when an organism is heterozygous, where both of these two alleles are interacting together, the recessive allele has no noticeable effect on the appearance, yet it's still part of your genome. So since my mom has blue eyes, I am heterozygous for eye color, most likely. And so even though I do not have blue eyes myself, if I have kids, there's a possibility that my kids might have blue eyes because I'm still a carrier, even though I'm not an expressor. Okay, so I'm heterozygous, and maybe you've heard of this before in the context of like Punnett squares. So we'll do an example of that next. So just to go through, how this is actually noted by geneticists. Okay, so the phenotype, of course, is the physical expression of all this, but the genotype, in this case, and the genotype for petal color in this example, is the combination of these two alleles. Okay, so if the organism, in this case, the pea flower, is homozygous dominant, homozygous means it has the same version of the allele, and both of those alleles happen to be the dominant type. That is homozygous dominant. So purple flowers, the purple allele is the dominant allele over white petals in pea plants, at least for the species of pea plant. And so an individual pea plant that has purple flowers and both of its alleles are purple flowers for flower coloration, we call that homozygous dominance. But let's say an individual has an allele for purple flowers from one parent, but also an allele for white flowers from another parent. It's heterozygous then because it has one type of each allele, and yet because it is heterozygous, the dominant one is the one that expresses itself. And so in terms of the phenotype, you see purple flowers. If, however, an organism has is homozygous recessive, it has the same version of the same allele, but both of those alleles are recessive, that's when you actually get phenotypic expression of the recessive trait, in this case, white flowers on on Mendel's pea plants. And so this is exactly what he did. He kept track of phenotype after multiple generations, having known what the parents were, because he was the one that was intentionally crossing them with his little paintbrush and keeping track of the data. So this, just to name some human examples, dominant traits can be things like freckles. So if one of your parents has freckles, odds are you have freckles. Widow's peak, so that's your hairline. If one of your parents had a widow's peak, odds are you have a widow's peak. The free earlobe, so take a look at your ears. If your earlobe kind of just floats free and you can wiggle it, that odds are if one of your parents had that, odds are you have that. Versus recessive traits. This is kind of interesting. We kind of think of freckles as being not that common because it's an uncommon dominant allele. Most people do not have dense freckles like that. So most of us are recessive expressive in this case. But a recessive trait, no freckles, a straight hairline, or an attached earlobe, where your earlobe literally attaches to the side of your head. So lab this week will actually be asking you to look at your own body and think about what alleles you have. So Punnett squares, and I'm almost done. I know that I'm already getting long in the tooth here. This lecture usually takes a while, so I'm going to break it up just to spare you too long in one recording. Punnett squares are a really handy way that people have devised to predict the results of genetic crosses when you have different parents. And this is basically what Mendel did, although he didn't devise the use of these Punnett squares. But it's really easy. If you know what the genotypes of the parents are, all you have to do is array them in this cross here, and you can predict what the resultant genotypes, and as a result, probably the phenotypes of the offspring are. So for example, here on the left, big letters denote, uppercase letters denote dominant alleles, and lowercase letters denote recessive alleles. And so in this case, the father or the mother, take your pick. This is homozygous dominant, this particular parent, versus this parent is homozygous recessive. Okay, so when these two cross and you simply do the Punnett square, all of the offspring are going to be heterozygous. It's that simple. And because they are heterozygous, they're pretty much all going to be expressing the dominant allele here. Let's go through a real example. I'm fairly familiar with this example because one of my buddies, this was his PhD dissertation. And so Tim, maybe you saw some pictures of him in the very first lecture where he was holding the fungus. He's such a geek. He likes fungi. Anyway, he did this pretty interesting, his PhD, this was part of his PhD, where he was studying monkey flowers and these two populations of monkey flowers, to be precise. Remember, a population is a group of the same species in the same place at the same time. So he studied these two populations of Diplodocus guttatus, which is, let's just call this the yellow monkey flower. And he had these, the same exact species that look pretty similar to the naked eye, but one was from the mountains and one was from the coast in California. And if you looked at these two different populations underneath a UV microscope or UV light, like what a bee would kind of see, for example, you noticed a different color pattern in terms of the UV radiation. The population A here, the front petal was all dark, whereas in population B, only the middle of the flower was dark. And so Tim, being the person he was, he decided to name population A runway population because it kind of, it's, you know, the whole petal. It looks like a kind of like a big runway if you're going to land a plane right here in the center of this runway. Versus this population B here, again, the same species, but different population. He called this the bullseye population for obvious reasons. Okay, so he was interested in what is the basis of the difference between these two populations of the same species. So he went and collected seeds from these two populations of monkey flowers, and he brought these seeds all back to a common garden in a greenhouse. And he basically did exactly what Mendel did. He did artificial crosses where he grew them up to maturity and he artificially crossed them. And when he did this, what did he do? What did he get? In the first generation between these two populations of monkey flower, he got all runway flowers, the offspring. Why? Because it turns out the parents were homozygous. In other words, the individuals from population A, the runway population, that was homozygous dominant, and it turns out the individuals from population B were homozygous recessive. And so the runway allele was the only allele that was expressed in the offspring, the F1 generation, because of this tendency. You can see this by the Punnett square here. And yet you don't want to stop there. That's kind of boring. The result, okay, I crossed these two different populations and I got one phenotype as a result. But then Tim he basically did exactly what Mendel did. He grew the F1 generation up to maturity, and he got their seeds and he planted them again and he artificially crossed them, and he got something entirely different because the offspring were heterozygous themselves. And if you do a cross of heterozygous offspring, you get something entirely different. You get some proportion of the F2 generation are homozygous dominants. Some proportion, 50%, are going to be heterozygous. And then he actually got about a quarter of the F2 generation actually expressed, again, the homozygous recessive allele. And so it's very consistent with what Mendel found. And Punnett squares allowed him to predict this quite neatly. So just to recap, we've talked about 3 possible genotypes from the combination of just working with 2 alleles here. We can have a homozygous dominant, which is denoted by 2 uppercase letters. We can have heterozygous, which is denoted by 1 uppercase letter and 1 lowercase letter. Then we can have homozygous, which is denoted by 2 lowercase letters. And we've talked about these 2 phenotypes in the context of these monkey flowers, runway and bullseye. And is this always the case when crossing different phenotypes, or genotypes rather? In other words, does this pattern always occur across all life everywhere? Are these traits that these Mendelian laws 100% manifest all the time? I hope you can predict my answer here. The answer, as it turns out, is no, because life is complicated and it does not always perform the way we expect it to. Oftentimes, Mendel's hypotheses are still very valid and very valuable, but there are a bunch of ways that inheritance patterns do not follow expected Mendelian ratios. So actually, I'm just going to go ahead and finish here because it's not going to be too much longer. And so there are a bunch of ways that organisms can have inheritance patterns that are not Mendelian at all. That if you try to use a Punnett square to predict what the offspring are going to look like on the basis of their phenotypes and genotypes, it won't work at all, because their modes of inheritance are not Mendelian. Homozygous and heterozygous allele combinations we've been talking about are a little bit thrown out the window. Okay, so let's just really quickly go through some examples of this. Heredity is not that simple. So for example, you can have something called codominance between 2 different alleles. And this is basically exactly what it sounds like. where both alleles refuse to give up any ground. The alleles are really stubborn. Neither will be recessive to the other. Okay, it's kind of like our political parties right now. No one will listen to anyone else. Okay, so you get co-dominance between two different alleles. On the one hand, was first studied in these camellias. Okay, so when you cross these two camellias, the white camellias and the red camellias, you don't get like red camellias in the first filial generation. You get something that looks like this, where both are fully expressing themselves and neither is dominant or recessive. Certainly not. Certainly not a Mendelian characteristic. This is not what Mendel would have predicted, and you wouldn't be able to predict this from a Punnett square, codominance. Another is incomplete dominance, where one specific allele is not expressed over its paired allele. So it's like kind of the opposite, where neither, where codominance both insist on being dominant, and in complete dominance both want to be recessive. And so you get this kind of mixing, is like entirely third phenotype as a result. So for in this case, this is a generic example: a white flower, mixed with a red flower offspring, and you get pink flowers as a result. It's literally like a mixing. It's creation of a third phenotype. It's a combination. So both are kind of being recessive and dominant at the same time. Not what Mendel would have predicted. Another thing is that sometimes genes are linked on the basis of where genes are located on the chromosome. Okay, so here is a chromosome right here, and a gene is simply a stretch of DNA along the chromosome. And so here's gene 1, gene 2, gene 3, gene 4. Some genes, because they're located really closely on the chromosome, they tend to be inherited together, because through the process of meiosis, when an organism is going from diploid to haploid, and it's splitting the genetic information, the genetic complement in half. Because these genes are located so close together on the chromosome, they're inherited together, even though the DNA is being split in half. And so these are called linked genes. So for example, kernel color and waxiness in corn are almost always inherited together. Okay, so certain varieties of corn, let's call them yellow, are almost always waxy, and yet purple varieties of corns tend to be a little more brittle. I'm making that up in terms of the color and combination there. But the reason why different colors of corn vary in their levels of waxiness is because the genes that code for waxiness and kernel color are linked. Okay, so they're inherited together. Okay, they're always inherited together. But wait, it gets even more complicated than that, folks. If these patterns of linked genes or incomplete dominance or codominance don't get complicated enough, they certainly do not conform to Mendelian inheritance patterns. It can get even more complicated, where one gene does not always even correspond to one trait, which I've led you to believe until now. And so you can get these different forms of non-Mendelian inheritance where you just don't know what the heck is going on. It gets even more complicated. So just to quickly go through some examples, you can get situations where one gene does not necessarily correspond to one trait. In other words, one trait could be the result of many, many genes. Okay, so for example, skin pigmentation. And I'm not referring here in terms of race, I'm referring in terms of tone. The genes that code for your skin tone, it's not one gene that codes for that. It's a heck of a lot of genes. It's polygenic. It's really, really complicated. Hair color as well is polygenic. There's lots of different genes that all kind of converge together to create those specific traits. Whereas some genes really are monogenic. Your hairline, for example, whether you have a widow's peak or a flat hairline, it's literally just one gene coding for that. Okay, so you can have polygenic inheritance, and certainly not Mendelian. Mendel had no idea that this was going on. Another example is this thing called epistasis. And epistasis literally means standing over in the Greek, which translates to some genes are bullies. Okay. Here's, on the bottom here is the easiest example to point to. Have you ever grown foxglove in your garden at home? It's a pretty common garden plant. Look at these different foxglove flowers. They're poisonous, by the way, so don't eat them. Sometimes dogs eat them and have to go to the vet. But look at these different colors of foxglove flowers. You'll notice that one is white, one is pink, one is kind of reddish, but they all have this spotted throat, right here in the middle. But why are this spotted coloration? Why does it only occur here in the throat of the flower and not throughout the whole flower? It turns out that this is a result of epistasis, where the gene that codes for petal coloration is dominant, it kind of bullies the gene that codes for this spotting. And so it more or less constrains the spotting gene to the throat of the flower as opposed to the whole, all the flower area. That is epistasis, is where one gene, we're not talking about allelic expression, we're talking about genes interacting with one another, and sometimes genes can bully one another. So in other words, this is a dominant gene, not a dominant allele. Okay, so epistasis is another thing that can lead. Gourds, for example, that you can buy around this time of year at stores are, this is all the same plant, and yet you get all this diversity of form and color because of epistasis. Another thing is pleiotropy, which is exactly the opposite of polygenic. Pleiotropy is when one gene can have multiple effects on multiple different traits. Okay, so a single gene can maybe affect trait A, B, or C. So seed shape, or pod shape, or flower color, or seed color, or pod color, or stem length. Some of those can all be affected by one gene. Some genes, not all genes are created equal. Some genes have an effect on multiple traits rather than just one. So it's kind of the opposite of polygenic, where multiple genes all affect one trait. In pleiotropy, one gene affects multiple traits. Okay, and this again is something that is still being studied. It's not resolved. Geneticists still bicker with one another about how this works. And the last example that I'll share with you, and this as an organismal biologist myself, I'm not a geneticist. I'm an organismal biologist. I'm relieved that this is the case. Is phenotypic plasticity, which is that the phenotypic expression of an individual plant or organism is dependent on the environment, on the organismal level, that the genotype does not necessarily have the final say of what the phenotype is. The organism's environment can have a pretty, oftentimes does have a pretty wide impact on phenotypic expression. It's a little bit common sense, but there it is. So for example, in this pretty elegant experiment here, you can take all this same species of plants, but you can of seeds, but you can plant them in different types of soil, compost, which is like really nutrient dense, regular soil, which you can get like just sitting in your backyard, and sand, which is really nutrient poor. And then you can do it even further by kind of just keeping it normal that you'd find out in nature or whatever, or you can autoclave the soil before planting the seeds in it, which is basically an autoclave is like a high temperature, high pressure oven that cooks all microbial life out of everything and makes it completely sterile. And so, you've basically got 6 different treatments here. This plant, these seeds were, natural compost, natural regular soil, natural sand versus natural or autoclaved compost, autoclaved soil, and autoclaved sand. And look at the result. It's all the same species, even probably same package of seeds. And yet look at this difference in phenotypic plasticity. Think of plasticity as something that's malleable. Okay, so this is not the result of genotype. This is the result of the different environments that these different seedlings are finding themselves in. Okay, so last slide here. All of this leads to something. Okay, why does all of this matter? We've talked about new alleles that arise due to mutations. We've talked about how sexual or genetic variation can occur through meiosis and recombination and mutations. We talked about how these things can be passed on to the next generation, whether that be Mendelian-like in a very predictable inheritance pattern or non-Mendelian in a non-predictable inheritance pattern. We've talked about how different phenotypes can emerge over time as a result of different environments they find themselves in. Now what? The end result of all of this, and if you apply massive amounts of time to this basic premise, is evolution, which is simply change over time. And we've already talked about evolution in some detail already. And so I've just justified on a molecular basis the end result, which is evolution. Okay. And with that, the course is over. No, I'm just kidding. Anyway, that's it for this tonight, folks. Thanks for hanging in with me. For those of you who are here, with that, I'll sign off. And do please let me know if you have any questions. And keep in mind, the Analyzing My Phenotype lab is this week. But with that, I'll see you on Wednesday for a discussion, a relatively brief discussion, I might add. I'm going to keep that short intentionally on DNA structure and function. All right, have a good evening, folks.